one of the most important sort of hobbies that Americans, it's not just Americans, people all over the world have, is tracing family history, searching for your roots. And I like to put this up because Time magazine back in the 90s had, on the cover showed that tracing family history, genealogy is America's latest obsession. It's really simple. Clicking your computer, it's easy as 1, 2, 3 clicks. One of the biggest companies in America is a DNA, I'm not a DNA, but a ancestry search database called Ancestry.com, which there are many people who have membership in that and provide, who have membership in the database who get access to the data in the database for searching family history. Now, this is big bucks. If you think about it, genealogy searching is number 2 in terms of internet businesses. If anybody know who number 1 is? Okay, I'm not going to say what it is, which I'm sure you all know. One of the groups that has the biggest interest in trying to understand family history and tracing roots are African Americans. A recent poll reflected that 80% of African Americans feel that it would be scientifically important to determine ancestry using DNA testing. And it's mainly because of the history of Africans in America, right? So the transatlantic slave trade, what some call a Maafa or a Holocaust of enslaved Africans from West and Central Africa, created a lot of lost identity in Africans in the New World. In fact, family history was lost, languages were lost, customs and traditions were lost. And so as African Americans, there's this very strong interest to sort of trace family history beyond the antebellum South across the Great Ocean into Africa. And so genetics has been quite helpful and useful in uncovering some of these histories. And we now, due to the genetics, can answer some very important questions. Now, this is what I find fascinating because genetics and DNA testing actually creates an interesting tension. It's the science and technology intersecting with society in many new, novel, interesting ways, and it creates a little bit of discomfort because we have to reconcile sometimes some issues like group membership, identity, how do we define ourselves? And we know that these definitions or these classifications change through time. And so now with the genetics, it's creating an even more tension that I find to be very interesting because it's allowing us to really reflect on what it is to be one of the so-called racial groups in the United States. Because for the most part, we all are mixed and we have very diverse ancestries. So because of the International Human Genome Project, we can now use DNA to say something about human evolution and population history. So what is it about this genome that's so interesting, so important? Well, scientists all over the world were involved in sequencing the human genome. There's 20,000 genes that make up the genome. In fact, when I was in graduate school, which wasn't that long ago, we were told there were 150,000 genes in the human genome. And then my second year of graduate school, they said 80, then it went to 60, then it went to 40, and then it was, oh, we only have 20,000 genes. And so folks were a bit shocked that they were not as far in terms of the human species from the nematode, which is a worm. 3 billion nucleotides, those 4 chemical bases, A, C, T, and G. And 23 pairs of chromosomes. And the fascinating thing about this is that we get half of our DNA from our mother and the other half from our father. They themselves from their parents. And so you share 25% of your genome with your grandparents. That is what we call ancestry, genetic ancestry. It's reflective of family history, shared genetics, and we can map out these lineages and look for these shared pieces of DNA that are in this mass genome of 3 billion nucleotides. Now, after we sequenced the human genome, we then went on to other organisms. We went to the rat, the mouse, which are models for disease, different mouse and rat models. So understanding their genome was important. The dog genome was important too. In fact, I kid guys all the time. The only other mammal that naturally gets prostate cancer besides the human male is the dog, the male dog. No other mammal naturally. We can, in the laboratory, get rats and mice we can induce prostate cancer in those animals, but they don't naturally get it. So what is it about the dog and the male, the human male? I don't know. There's always an interesting story with that. So when we sequenced the dog genome, we found that there's about 19,000 genes in the dog genome. 19,000. So really, there's not a lot that separates us from our best friend, and in fact, that's why some of us resemble our pets. Those 20,000 genes are on 23 pairs of chromosomes. Some of them are bigger, so we call those gene-rich. Some are smaller, and we call them gene-poor. Many of those genes actually are involved in susceptibility for disease, like breast cancer or prostate cancer, type 2 diabetes, hypertension. And in fact, growing up, I'm sure your mother or your father may have said something to you like, "You act like your daddy," or "You look like your daddy." And I would say to my mother, I hope I do, yeah. I should resemble my father. I share half of his DNA. But those genetic signatures that we inherit, some of them actually are involved in risk for disease. And so that's the big challenge. And what gets me up in the morning every day is to really try to understand how the transmission of particular genetic variants impact the transmission of cancer. So what are these genetic variants? We call those polymorphisms. Poly means many, morph means form. So we have many different forms of the DNA. So if we look out in this room, there's maybe a couple hundred people in here. There'll be many different patterns in the DNA if we compare any two people. In fact, we've cataloged over 40, 50 million of these polymorphisms. A class of polymorphism is called SNP, a single nucleotide polymorphism. So let's say we're looking at a track of DNA, ACT, CAG, TTCA. Maybe 94% of you guys in the room may have a C at that second to last position, while about 6% may have a T. That's a polymorphism, a SNP, single nucleotide polymorphism. A subtle change, and we've cataloged millions of them throughout the genome. Some of them have detrimental effects on the gene, and like sickle cell disease, it's due to one polymorphism in the beta globin gene. Others, we need several, other diseases are due to several of these polymorphisms. They're more complex. So that's sort of the job that I have, is to try and understand how the inheritance of these polymorphisms may impact the susceptibility for disease. Now, those SNPs actually reflect history. They're like tags. They're like markers. And just like if you're a genealogist and you're searching in your family tree, you have these markers that reflect time periods and who married who and how many children they had. These polymorphisms are useful for tracing family history too. Think about it like this. We inherit them from our parents from their parents, and we can then trace within a family these genetic variants, but also within a community, and those communities within continents, and it would reflect overall genetic ancestry. So when we talk about continental variation, what do we see, pretty much? We see that most of the variation is structured in Africa. There is enormous genetic variation in Africa compared to other continents, Europe and Asia. And it's mainly because humanity started in Africa. If you guys didn't know, modern humans evolved out of Africa about 100,000 years ago. They left about 80,000 years ago, went into the Middle East. And when they left out of Africa, they carried with them a subset of that genetic variation that's rooted in Africa. And so when you look at this Venn diagram, you see that the circle is bigger for Africa compared to Europe or Asia. There's a lot of overlap, right? That overlap reflects the common nature of humanity. We all have shared genetic variants, right? Because we're all humans. We're all one species, right? But there are also some polymorphisms that are restricted to particular continents. And this is important to understand because it allows us to say, okay, this profile reflects one of African ancestry, this one reflects one of European, and this one reflects one of Asian. What I find fascinating is that 50% of the genetic variation, if you look at those numbers, in Africa are exclusive to Africa. That's because humanity started in Africa, a subset of individuals left out of Africa and peopled the world, while only 10% of that variation in Europe, 10% in Asia are exclusive to Europe and Asia. So if you are a scientist and you're trying to understand risk for disease and drug response, you really want to know all the variation that exists in your population, that exists in America, let's say. And so you really have to not only look at people of European descent and Asian descent, but also African descent, because that's where the bulk of the variation is. But the fun part is this: we can actually look at these DNA profiles and trace the movement or the migration of modern humans. Remember what I said, out of Africa about 80,000 years ago into the Middle East, going into Europe about 40,000, into Asia about 50,000, Southeast Asia about 40,000, and then across the Bering Straits into the New World 15,000 years ago. We can see that in the signature of the genomes of these populations that are descendants of the common ancestors that migrated out. This is cool because what it says is that there are no distinct sort of human races biologically. We're all sort of mixed at some level, but we do share, and we do share a common ancestor, but we also have more recent ancestry with people who lived closer to us. So the closer you live together, the more genetically similar you're going to be. So here we are now in the United States. We're using self-identified race, right? The government, they always ask you, white, black, Asian, you have to mark on your census documents, you go for insurance or your driver's license. Black, white, Asian, Native American, Pacific Islander, American Indian. It emphasizes the geographic region of origin of a person's ancestry, which is cool. However, when we think about African Americans, right, there is a lot of mixture. We have a wide range of variation in the African American population. We go from Halle Berry or Vanessa Williams to Wesley Snipes. That's the guy in the middle there. That's the full range of the African diversity. It's quite diverse and reflective of different family histories and genetic ancestries. I like to put this up because sometimes people don't really appreciate or understand the amount of genetic variation that's within the African American population. Very heterogeneous, very diverse. And then I put President Obama up there too, because in the US, he's considered black. However, genetically, he's very dissimilar to the other African Americans on that page, because his father is from East Africa. He was not a descendant of enslaved Africans that came from West Africa. And so genetically, he's very, very diverse. Now, I tricked some of you guys. Some of you guys thought that was Denzel Washington or something, but that's Vijay Singh. And he's not African American. He's of Indian ancestry, so he has Asian ancestry. And in the United States, race is mainly based on skin color. And so if you saw him from a distance down Halstead Road, you'd say, oh, that was a black male, when in fact, he's Indian. And so because skin color is this definitive sort of marker for race in America, we always are trying to reconcile these contradictions.